This cost-effective method can find wide applications in molecular diagnosis and screening of FXS and Fragile X-related disorders, with faster turnaround time and less investment in equipment. Our PCR-based assay can facilitate classification of full spectrum of the FXS and Fragile X-associated disorders, including intermediate, pre-mutation and full mutation, with robustness and rapid reporting time. The procedure will be demonstrated by Wang from Perkinelme, Singapore.
Start by removing the PCR buffer mix, sample diluent, and DNA samples from the -20 degrees Celsius freezer and leaving them at room temperature for 20 to 30 minutes to thaw. Prior to using the reagents, vortex and briefly spin them down. Measure the DNA sample concentration with a spectrophotometer, and if required, dilute it to 25 nanograms per microliter with sample diluent. Label the wells of a PCR plate to identify reference and tested DNA samples, and calculate the number of reactions needed for the test, reference, and negative control samples. Prepare the PCR master mix by combining 15 microliters of PCR buffer mix, 2.6 microliters of sample diluent, and 0.4 microliters of polymerase for each reaction. Vortex the mix and spin it down. Then dispense 18 microliters of the mixture into each well. And pipette 2 microliters of each.